I'm going to now introduce our final speaker of the scientific session before we move on to our next keynote. Taras is coming from the Ari Kavetsky Institute of Experimental Pathology in Ukraine. And Taras is going to talk about the expression of collagen density regulating factors in prostate cancer. So good to see you this morning or this afternoon, Taras, and looking forward to hearing more about your work.
Okay. Hello, everyone. Thank you for a chance to present our classical short study. It's a typical histopathological study, and today we talk about prostate cancer and about expression of collagen density regulation factor. As you know, prostate cancer is the most frequently diagnosed cancer and the 5 leading cause of cancer deaths among men in 2022. The molecular and cellular mechanism underlying the development of prostate cancer are not yet completely understood, and it is known that the tumor microenvironment play a key role in the progression of many neoplasms, including prostate cancer. And tumor microenvironment consists of tumor cells, tumor stromal cell like stroma of fibroblast, endothelial cells, adipocytes, stem cells, and innate and adaptive immunity cells such as T and B lymphocytes or macrophage dendritic cells, natural killer cells, neutrophils, amyloid islet suppressor cells, and etc. These cells are laid on the scaffold formed by the extracellular matrix, which including proteins, proteoglycans, or glycoproteins, and chemicals secreted by cellular components such as growth factors, cytokines, chemokines, microRNA, and other. Tumor cells interact with other cellular components, and the extracellular matrix provide a structure to hold all this element together. Interaction between cellular immune cells and non-cellular compound account of the tumor heterogeneity and complexity of tumor microenvironment and limitation for target therapy in patient. While the role of cellular component of the tumor microenvironment is currently relatively well characterized, the role of extracellular matrix and its remodeling during the tumor process remain poorly understood. Disruption of the extracellular matrix play an important role in the development and progression of many malignant neoplasm, and the composition and structure of extracellular matrix are unique to each tissue and vary according to its function. The interaction of tumor cells and which the extracellular matrix affect a number of biological processes such as proliferation, migration, invasion, or metabolism. Normally, the extracellular matrix is required to maintain cellular architecture and polarity, which is disrupted during neoplastic growth. During carcinogenesis, the extracellular matrix is actively reconstructed, and as a result of the remodeling, change in the composition and structure of the extracellular matrix contribute to normal behavior of the stromal cells, in particular fibroblast and immune cells and the endothelial cells. This is why the future of extracellular matrix are currently considered as prognostic and diagnostic markers, and matrix itself as a new therapeutic target. In our previous stage, we create algorithm for analyzing and analyzing the quantitative characteristic of the collagen matrix of different neoplasm, which and you can see this algorithm in screen. But we select to selectively determine collagen in tumor tissue. We used classical histochemical methods like Masson staining, FFP tissue block, and after on the microphotographs we on the obtained slide, image separation was performed using the Photoshop program, and morphometric analysis was performed using the CurveLive program. It's very similar program, but first times this program create for analyze SHG microscopy photo, but we use classical image with light microscope, and after the density of collagen in the tissue was determined as the ratio number of collagen fibers measured using CurveLine program tool. This area obtains then working with the certifier model program, and obtain with the ImageJ program. We found it, and it's our preview result, but we found the prostate cancer tissue had two times lower collagen density and 1.3 lower of collagen alignment compared with benign prostate hyperplasia tissue. In prostate cancer tissue, collagen fibers was shorter and thinner, and moreover, in other study, we found that tissue of metastatic prostate cancer was characterized by a 2 times higher density of collagen matrix compared to non-metastatic prostate cancer. And there was not a significant difference in the alignment indicate in the study group, but we found that the length and width of collagen fibers in metastatic prostate cancer tissue were significantly lower than in non-metastatic tissue. And now our investigation aimed to study the expression of collagen density regulating factor in prostate cancer. As you can see on the slide, our work is based on the analysis and the result of the treatment and examination of 40 prostate cancer patients who were treated in the National Cancer Institute in Kyiv, in Ukraine, and determination of collagen in the tissue of prostate cancer was carried out by histochemical method, mesotelioma staining. Morphometric study of collagen fiber organization were performed using ImageJ and CurveLine programs, and based on the result of our morphometric analysis, we divide all prostate cancer sample into 2 groups with low and high collagen density. In the first stage, we analyzed the expression of transforming growth factor beta, or TGF, which is a key factor that modulate the expression, modulate function of tumor-associated fibroblast, and its a cell who that the main source of both collagen and matrix metalloproteinases, what involved it collagen structure regulation. And as you can see, prostate cancer tissue with high collagen density is characterized by significantly higher TGF beta expression level. Collagen cross-link and deposition depend on lysyl hydroxyproline, which is catalyzed by procollagen lysine 2-oxoglutamate 5-deoxygenase or plot. Aberrant lysyl hydroxyproline and collagen cross-link contribute on the progression of many collagen-related disease, such as cancer, and expression of plot is regulated by multiple cytokines, transcription factors, and microRNAs. This regulation of plot promote cancer progression and metastasis suggest that targeting of plot is potentially strategy for cancer treatment. And we found that prostate cancer tissue with a high collagen density is characterized by higher plot expression level. On the next step, we study expression lysyl oxidase in prostate cancer tissue. It's enzyme responsible for initiating covalent cross-link formation in collagen fibrils, and this enables the formation of cross-links between telepeptides and the helix of collagen molecules, suggested by 4D period axillary. During maturation, these cross-links interact, thus forming trivalent cross-links. Cross-link formation is essentially determinant of the property of developing and mature connective tissue. And you can see what prostate cancer tissue with a high collagen density characterized by high level of expression of this enzyme. On the next step, we study expression of serine protease inhibitor or serpin E2. Its molecule can bind to the extracellular matrix on the surface on fibroblast or several other cells, and serpin E2 form complex with certain serine protease like urokinase type plasminogen activator, tissue type plasminogen activator, plasmin, and trypsin in the extracellular matrix. And we found that the prostate cancer tissue, the higher level of serpin E2 expression in prostate cancer tissue with high collagen density. And so in the next step, we study expression of matrix metalloproteinases. Two type, it's collagenases and gelatinizes. Gelatinizes, it's type MMP 2 and 9, and collagenizes, it's MMP 1, 8, and 13. And we found, as you can see on the slides, of prostate cancer tissue was characterized by a significantly higher expression level only of MMP 13. And to summarize, the obtained data indicate the involvement TGF-beta, serpin E2, lysyl oxidase, and plot in regulating of collagen matrix density in prostate cancer, and suggest the potential role in prostate cancer metastasis. Thank you for attention.